so that's exciting. Today is one of those things that I wish I didn't have to point out still, and I hope we're trending in the right direction. But today is another one of those exciting days where we have seminars by two female grad students who are mentored by two female faculty. And so I just want to call attention to that and say, hopefully we look forward to the day we don't have to call it out, but it's a great thing to see it happen. So without further ado, I'm going to turn this over to Caitlin Pepperell to introduce Jessica.
Thanks. So I'm going to introduce Gardnerella vaginalis and Jessica, and I'm going to start with the bacteria. So my lab works a lot on Mycobacterium tuberculosis, which is slow growing and fussy and fastidious and kind of awful. And I didn't think that we would find a bug that was worse, but Gardnerella is worse. So just keep that in mind as you listen to Jessica's talk. So Jessica started in the lab in March of 2020 without any experience in microbiology. I gave her this project to work on, which is with this terrible bacterium, and was a project that had bedeviled multiple lab members over many years. So this bug is very difficult to grow. It's fussy. It just decides to die. I can see Vivian nodding, who has direct experience. And yeah, it's just generally kind of a jerk. And the goal of this project was to create an experimental evolution system in the lab whereby the populations were maintained during serial passaging, which was a very difficult problem and had vexed very experienced microbiologists. So you can imagine this was a serious challenge for somebody new to microbiology in the setting of a pandemic and trying to sort through these problems in an empty lab. And Jessica has done a wonderful job, and I'm very excited and proud to hear about the results that she's going to tell you about today. Thanks.
All right, can everyone hear me? Yeah. Great. Thank you for coming out today, and thank you, Dr. Pepperell, for the very flattering introduction. So this is the first time in about 2 years that I am able to present my work because of COVID and other circumstances, as I'm sure a lot of the other presenters have had to experience this time around. So because of that, I'm very excited to talk about my project, as Caitlin has said, and just show you what we have accomplished with this project, as well as the many future directions that we can go with this. If it will go. Oh. Maybe if I click on it manually. Ah, there we go. All right, so I thought the best way to start off my talk is to, well, introduce the lab. Caitlin's already done a good job with that, but I will also introduce the lab. So I'm part of the Pepperell lab, which is a part of the microbial sciences building. And the main goal of our lab is to investigate the evolution of bacteria and see how bacterial adaptation contributes to the pathogenic nature of bacteria. So we study 3 species of bacteria in our lab. As Dr. Pepperell said, most of our work concentrates on Mycobacterium tuberculosis, where we have students that are evolving pellicle biofilms in order to look at the different phenotypes those biofilms can make, as well as seeing if they incur any type of drug resistance. We also work with this bacteria here, Staphylococcus saprophyticus, which is the second main cause of UTIs behind E. coli. And we are looking there at how it has jumped from the environment to humans in order to cause disease. But as Dr. Pepperell mentioned, I will be talking about this bacteria right here. As mentioned, it was a very finicky project to get off the ground, but it has gone off the ground in the past couple of years. So I'm really excited to talk about it today. So Gardnerella vaginalis, or GV, as I will be referring to it from now on, is a gram variable rod-shaped bacteria. The reason I say gram variable is that the cell wall of the bacteria is so thin that on a gram stain, it can stain positive or negative. So if you look at this bottom left corner, this is a vaginal epithelial cell with garnerelle attached to it. The projector isn't really showing the colors very well, but you can see there are dark purple and light purple cells. That is the garnerelle on the cell being stained as either gram positive or gram negative. It is also a facultative anaerobe, meaning that it can survive in oxygenated and non-oxygenated environments, which is actually really important for its survival in its host niche. It loves to interact with other bacteria, as we found in both our studies and in literature research. The literature research has shown that GV can actually enhance the virulence gene expression of other bacteria within the microbiome, and those partners can also increase its own virulence gene expression. So it really likes collaborating with other bacteria in its environment. The last important thing you need to know is that it is a niche specialist, in that you will not find this bacteria in the environment, you will not find it in other animals, but you will only find it in humans, and it mainly resides as an active member of the vaginal microbiome. So why are we studying GV in the first place? It happens to play a role in the development of a disease called bacterial vaginosis, or BV. So BV is a common reproductive disease that affects women of reproductive age, and GV is usually considered to be the causative agent of this disease. And it's not like your typical bacterial disease where, you know, you get infected with a bacteria, it comes into the body, and the body doesn't recognize it and it kills everything. This is kind of unique in that GV is already a naturally occurring part of the microbiome. So this was taken from a study that studied the microbiomes of healthy women versus those who had BV. So the healthy vaginal microbiome is made up primarily of lactobacillus, such as L. inners and L. crispidus, as well as others. And the starred part here in the purple is Garnerella vaginalis, and this is, it kind of resides in the minority of the biofilm. However, they found that in women with active BV infections, you can see that there's a drastic reduction in lactobacillus, as well as a drastic increase in GV. And this essentially stands for a shift in health status of the microbiome from a healthy to an unhealthy state. And this is what we call a dysbiosis. And we believe this is how BV has the ability to cause infection. And if BV is able to get severe enough, it can be associated with the increased risk of preterm birth in women of reproductive age that get this infection. And it's also associated with an increase in the acquisition of STDs. So another pesky thing about this bacteria is that it has the ability to form this very stable structure called a biofilm. For those of you who may not know what a biofilm is, it's a structure that bacteria create in order to hide from essentially a very stressful environment. A common conception of a biofilm you may know of is a dental plaque. That is essentially a microbial community that sits on top of the tube and interacts with the tube's surface and can even cause damage. So biofilms can be problematic in general in that they can incur resistance to antibiotics when there's shear forces such as blood, urine, other liquids in the body that may be trying to shoot an infection out. It makes it harder for the body to clear that infection. And it can also hide cells from the immune response. So that makes it even harder to clear the infection when biofilms are involved. This can create the presence of chronic persistent infections where you have the bacteria stick around for long periods of time and it may develop antibiotic resistance. So GV tends to play a very prominent, or it's been hypothesized to play a very prominent role in the formation of the biofilm itself. So many in the field have theorized that it actually acts as a scaffold to create the biofilm. So first, it adheres to the vaginal epithelium where it will start colonizing the cells and starting the infection. And then as it forms the biofilm, it will pull what we call late colonizers in this particular figure into the biofilm. And these late colonizers tend to be other anaerobic bacteria that may be present in larger amounts at the time. And then once it forms this mature biofilm, it's done what it's needed to do. It's housed the bacteria, it starts killing the host cells. It will release GV back into the environment and it will keep colonizing the niche and furthering the infection. So we mentioned earlier that we use experimental evolution many times in our lab. And you may be wondering, how can we use experimental evolution in this particular context to, see how GV forms a biofilm and contribute to the pathogenicity of this disease? So the crux of my work revolves around experimentally evolving GV populations. And we believe if we evolve different populations of this bacteria multiple times, it will reveal variants that may be involved in pathogenic traits such as biofilm formation. So in order to design this experiment, we decided to create an evolve and resequence experiment where we are experimentally evolving GV biofilms again and again. So for this particular experiment, I ran 3 courses of the experiment with 9 independently evolving GV populations altogether. And these graphics will show how I went about designing the experiment. So first we revive our ancestral strain of bacteria for about 1 to 2 days. It's growing planktonically in a tube in liquid media. And then after it is grown, I will plate the biofilms in these 96 plates where they can adhere to the bottom well, grow biofilm. And that happens for about 24 hours. So in this experiment, we want to select for biofilm cells. So how do we do that? So here is kind of what happens when a bacteria grows a biofilm. You'll see the biofilm in blue that adheres to the bottom of the well. And there are cells that kind of float on top that are not necessarily part of the biofilm. We do not want these floating cells. We want the cells that have made the biofilm successfully, that adhere to the plate and contribute to the biofilm. So what we do to select for biofilm cells is that we remove these cells that have supposedly been kicked down and don't contribute anything. We replace it with new media. And then we transfer a subset of the biofilm cells, dilute in fresh media, and then it goes into a new well or new plate where that biofilm can grow for 24 hours. So we repeat this process again and again for as long as possible until we see phenotypic change. During this process, I conduct a number of phenotypic assays to quantify the density of the biofilm at each step in the process. And I also freeze down samples so that we can later conduct whole genome sequencing, where we can compare evolved biofilm genomes to ancestral ones. So we have an assay that's looking at phenotypic changes that are happening through this process. And we also have an assay where we can look at what's happening at the genetic molecular level. So one of the main findings that we have found in this experiment so far is that when we evolve these populations again and again, the pathogenic strain that I'm working with, GB2, tends to make more biofilm in about 5 passages or so. So here's a visual representation of some of the phenotypic data that I have gathered. This comprises of all 3 experiments and all 9 independently evolved populations. I used 2 different variables to measure the biofilm density. So OD biofilm is essentially us just looking at the biofilm at OD 600. And crystal violet stain is what we use to stain the extracellular matrix of some of the biofilms. So this essentially stains the outside the biofilm and quantifies its mass in just a different way. So one thing I hope you can appreciate is that over time, we do see spikes in growth in basically all of our experiments. This tends to happen over the course of 4 to 6 passages. And so you can kind of see them in both of our variables and all of our experiments, that we do see an increase of biofilm growth from kind of this starting point here. What I also hope you can appreciate is that we have divergent trajectories that are occurring between most of our populations, and this can be due for a number of reasons. So first of all, these populations are evolving independently, meaning that they are acquiring their own traits and they're kind of following their own evolutionary path. So some may be evolving to create more biofilm, some may be evolving to create less biofilm. Oh, and by the way, these red squares are just me highlighting some of the times where they've diverged a little bit. There are a couple other reasons that this may be occurring. One of them is that the bacteria get clumpier over time. There's an increased amount of cohesion, which makes the spectrophotometer read the samples a little bit differently. Also, as Dr. Pepperell mentioned, this bacteria is incredibly finicky to grow. You can actually see instances where it kind of crashed over time. And so this could be a product of just the bacteria not really liking the model and just trying to adjust to this lab life. But this is kind of really interesting that we're seeing these divergent trajectories, and that's kind of what we're looking into right now. But overall, what we have found is that when we evolve these populations, we've frozen them down and we revive them and compare them to ancestral strain, in general, they create thicker biofilms, the evolved strains, than the ancestral strains. So this is just a visual, just a visual of the crystal violet stain. So you can see that the biofilms for the evolved strains are typically thicker, and they cover an entire surface of the well compared to the ancestor. We also took SEM pictures of our evolved biofilms. I'd like to thank postdoc in our lab, John Kearney, for this. He specializes in SEM, and he was nice enough to take pictures of these biofilms for us. So we compared pictures of the ancestral biofilm to our evolved ones. I don't have them all pictured up here. They're just kind of the few we took as representative samples. And you can see that there are some pretty drastic differences between structure of the ancestor and the evolved populations. You can see that there are some similarities in there's kind of like a lattice spreading structure that a lot of these share. But you can see in, simple terms, there are more clumps. The clumps seem to be bigger, and they are bound by what we professionally call sticky stuff. Just kidding, it's extracellular matrix, which is essentially the carbohydrates, the proteins, and eDNA that help to bind the biofilm together. So this is just a zoom in of the biofilms that I showed you before. You can see the scale bars a little lower. I believe this is 10,000x more into the biofilm. And this is just to show you what the extracellular matrix kind of looks like in the ancestor versus the evolved. You can even see that there's a little bit of structure difference between some of the evolved strains. So there may even be some variability between our evolved strains as well, which is interesting in itself. So I showed you, first of all, the phenotypic data from our experiment. Now I'm going to go into what we saw when we sequenced our genomes. So when we compared the genetic structure of the evolved biofilms versus our ancestral ones, we saw that there was convergent evolution of populations, evolution of variants in pretty much all of our biofilm populations. So just to give you a better look into the data we use, essentially why we did this experiment was to explore the heterogeneity of the evolved biofilm populations versus ancestral ones. We wanted to see if they became more diverse, if they became less diverse, or if there was anything that was potentially being selected for our populations. So among the 9 populations that we tested, we saw that about 2,000 variants in total were recorded. So new variants that came to light in our experiment. And out of the 2,000 variants that were recorded, about 50 of them recorded approximately a 30% change in allele frequency, which is a fairly drastic change. And it's kind of our first indicators to suggest that it's not just drift that's acting on these samples. Maybe selection is also acting on these variants. All right, so I mentioned convergent evolution before. I want to go a little deeper into what exactly we're looking for. So when we talk about convergent evolution, we're essentially looking for similar mutations that are appearing in independently evolving populations. So these samples aren't being mixed in any way. They are kind of evolving on their own trajectory. So here I'm just illustrating 2 examples that we're looking at when we're trying to look at convergent evolution. So think about for each population, there's a collection of genomes, and this is visualized by the different color ovals here. And the circles you see are different hits that we've gotten from our data that have these like higher allele frequencies. So for example, locus number A may be a gene region, and you can see that not all the hits are located in the same place, yet they reside at the same locus. So this could be that the hits are affecting different parts of the gene, or the locus, I mean. And it would be interesting to see if they are hitting different areas, but we still consider them convergent evolution because they are occurring around the same locus. For locus B, when we look at intergenic regions, we narrow our range quite a bit. I believe we kind of look for 0 to 20 base pairs, just to make sure that the hit that we're seeing is hitting the exact region, and who knows, maybe it's affecting gene expression or something else. So we got 2 striking hits from our data. The first one aligned to a type 1 fatty acid synthase in Gardnerella vaginalis. I unfortunately don't have a picture to show you of one in GV, and I'll explain that a little later. But this is a homologous structure of the type 1 fatty acid synthase in Mycobacterium tuberculosis. And this is just a general view of what it looks like, some of the catalytic domains that are involved, such as ketoreductase over here and ketosynthase, and just how the domains are structured in the protein. So when we found that there was, so I just want to say that there's a lot of activity going on in this gene. We found it in all 9 of our populations. What we wanted to see was how these mutations were operating within our samples and how quickly they rose to fixation, or, well, what exactly was going on. So one of the trends that we noticed in all of our samples is that they were experiencing positive selection. So essentially what we're looking for here is variants that rise very quickly to fixation or almost fixation within a short period of time. So this flat line essentially resembles one passage. So the ancestral and the 0 is one passage. So you can see that these alleles are not appearing after one passage, but after about 5 to 10 passages. Some of them will shoot up to about 80 to 100% frequency, meaning that they're present in basically the majority of the population. So this implies that it may be, these mutations may be genuinely affecting the structure of the fatty acid synthase, and that's something worth exploring. Another trend that we have seen happen in our populations is this appearance of secondary mutations. So you see there's usually one allele, this is all at the same locus, just want to point that out. There may be one mutation that's rising to high frequencies, and then you may see another mutation at the same locus that's also rising in frequency, which may be implying that these mutations may have additive effects to the fitness of the population. And we also have other populations that are experiencing clonal interference at this particular locus. And the best way to illustrate this is that there's usually a mutation that rises pretty quickly in frequency. I'm sorry, these graphs are so small. There's usually, this is a passage 5 and passage 10 in these graphs, I believe. So they tend to rise over the course of a few passages, and then this trajectory starts falling, and then another one starts rising. So they usually meet in the middle around 40 to 50% frequency. And this implies that the mutations are kind of competing to incur a higher fitness level in the population. So these are all very interesting trends that we've seen in our data, and we are working very hard to explore them more right now. So what exactly do these mutations mean? The short answer, but also the most difficult one, is we really don't know. We can only really hypothesize at this point. We do know that fatty acid synthase in general is highly conserved across many organisms, as fatty acids and their synthesis are essential to organismal survival. Fatty acids, at least in prokaryotes, serve as the hydrophobic component of the bacterial membrane. So we believe they may be involved in maintaining the permeability and the structure of the membrane. Also, with the mutations that we found that almost all of them were non-synonymous mutations, and they were found in these catalytic domains. So this is significant because this is an essential enzyme, and we're finding mutations that may be drastically altering the function of either the enzyme itself or these domains. And these are just specific domains that I listed over here. So given that we have this, we are theorizing that this may affect the lipid production altogether in the way of either maybe creating truncated products or basically affecting the rate at which the fatty acids are being made. Otherwise, we really don't know a whole lot, even about fatty acid synthase in general, and especially GB. And that is something we're definitely going to explore more in the future. So I don't know how much time I have to go through this, but we had another hit in our population, meaning that we found a SNP at an intergenic region that was located in about 3 out of our 9 populations. So this SNP was actually located in the different populations within 5 to 10 base pairs of each other. So essentially at the same location, they all seem to be experiencing positive selection trajectories in which they seem to be rising in frequency very quickly. I'd like to point your attention to this green one, which is kind of steadily falling. This was a population where this quickly rose to fixation. Now it's falling, but we also had other fatty acid synthase mutations that were rising to fixation. So it's possible that even this SNP is working, in conjunction with or competing with these mutations. So it'll be really interesting to see what exactly this SNP does for the bacteria and the biofilms in the future. So thank you, Dr. Perna, for reminding us to put this slide in. So I'm going to talk a little bit about rigor and reproducibility in our work. So for my particular experiment, we keep the growing conditions and the protocols very consistent between the experiments. They always grow anaerobically. They always are passaged in a certain way. We use proper aseptic technique to prevent contamination. And we change tips a lot so that we don't mix our populations so they're not being influenced by other independently evolving bacteria. We also back up all our data onto multiple places, whether it's a hard drive or Google Drive or wherever. And our lab has a shared electronic lab notebook system, so we can all access our lab notebooks if we're like picking up a project from someone or we need to see what someone else did. I personally keep very copious notes, especially because of how finicky GB has been. It's just really helpful to keep all those notes just to, make sure you know what's going on. So to summarize my work, first of all, I'd like to reiterate that GV remains a significant health concern to women of reproductive age, especially because it can cause all these severe side effects and risks for future diseases, and it can also become resistant to commonly used antibiotics. I've also shown you that when we experimentally evolve our populations, there's a potential to reveal variants that may be involved in biofilm formation, which could contribute to its pathogenic nature, potentially in disease. And I've also shown you that we have discovered convergent evolution of variants that in our evolved biofilm populations. However, a lot more research needs to be done to determine the function and the significance of these variants. So for the genic 1 and the type 1 fatty acid synthase, we want to find out how these variants impact the function of the enzyme. And then for the intergenic SNP, we also want to see if that variant has an impact on gene expression and how that's affecting biofilm formation. So there are a lot of different directions that we are going to try to validate these hits that we've discovered. First of all, we are obviously interrogating the function of these hits. For the type 1 fatty acid synthase, we have submitted samples to lipidomics where we can compare the fatty acid profiles of our evolved biofilm strains versus ancestral ones to see if production and structure of the fatty acids are being changed. We're also going to do RNA-seq in general, not just to see general expression changes, but potentially how this SNP is involved. We're also planning on doing allelic replacements. Mostly because we can't knock out the domains or anything because the fatty acid synthase is supposedly the only one and it's essential, so it could kill the organism. So we're going to try to replace specific residues or SNPs within the bacteria to see how that affects GV in general. Another thing we want to do is determine if these variants that we found are biofilm specific. So there are a couple of ways that we can do this. First of all, we can passage the ancestral strain, like start the experiment over, but instead we passage ancestral strain as a planktonic culture. So there's no pressure to grow as a biofilm. So if we have hits in both of these experiments, for example, that may be a sign that they're arising due to general lab adaptation and not exactly due to the biofilm selection pressure. We also are conducting a backpassaging experiment where we actually grow up our evolved biofilm cells, take away the biofilm selection pressure and passage them as these planktonic liquid cultures. And this would essentially show us if it refers back to the ancestral state, if it loses the mutations that is gained through the biofilm selection pressure, or if it starts, producing less biofilm. So there's a lot of directions we can go for this. I actually haven't listed all of them, but this is kind of what I can fit into in this presentation. So there are a lot of people I'd like to thank for the work I've conducted. First of all, I'd like to thank my whole lab, especially Maddie Toth, who is a graduate student in the MDTP program, who's been helping me keep the longer term experiments alive as well as with the bioinformatics analysis. I'd like to thank John again for the SEM pictures, and I'd like to thank Dr. Pepperell, who has been quite a wonderful mentor in these weird circumstances. It's not easy joining a lab, not just on your 5th rotation, but a week before everything shuts down. for a pandemic. So she has really helped with navigating those obstacles, and I couldn't be more grateful for her support. I'd also like to thank Tracy Smith, who was our former lab manager and who passed on all these protocols down to me to make this possible. I'd also like to thank our collaborators, Aaron, Amy, and Katie in the Dillard lab. So Dr. Dillard has been collaborating with us with GV work for a while, and they really showed me the ropes with how to work with GV when I had absolutely no microbiology experience and no one in the lab to help me. I'd also like to thank my thesis committee for their support and suggestions, as well as various people in the genetics department and my funding sources. And I will take any questions you have for me now. Thank you.
We are a bit short on time, but let's take one or two questions.
I have a question. So you mentioned at the beginning that it was a facultative anaerobe, right? That it could go in both like aerobic and anaerobic conditions.
Yes.
And you said that that was like important for where it lives in like its context, right?
So yes, one of the theories about the development of bacterial vaginosis, like I believe if I'm remembering correctly, the vaginal microbiome is mostly in an aerobic space. And when we don't know what the trigger is for these particular conditions, but it does seem to make the bacteria more able to thrive in an anaerobic environment. So when GV starts overgrowing and pulling in these anaerobic partners, they're becoming more apparent in the population. So it can survive in aerobic conditions and it can survive in anaerobic conditions. And also that's really helpful with making a biofilm, because a biofilm can have multiple oxygen and nutrient gradients. So these cells can survive at, at the bottom layers of biofilm when there's not a lot of oxygen, and also where there's not a lot. So this can be a very beneficial trait for an organism. So we have found that's the best way to grow this particular strain. And a lot of studies do grow it anaerobically. We grow it in an anaerobic chamber, which kind of sucks out the oxygen in the bacteria.
Are there plans to try growing it aerobically? Just like simulate that?
Not at the moment, but that is a really interesting suggestion. I do remember it wasn't as successful growing aerobically. We can always give it another shot, but we've just kind of been growing everything anaerobically to ensure the success of the growth of the bacteria. Like I said, it's a very finicky grower. It doesn't grow very easily. Thank you. Over here.
Have you done the experiment where you take evolved and you have to remember passages and then compete them with the ancestral strain to see if the selected variants actually do confer advantage in that?
So that is an experiment we are thinking about doing that is currently in development. We have not quite done it yet. I'm still developing the protocols and everything, but yes, we would be competing the evolved biofilm cells with ancestral ones. We would expect the evolved biofilm cells to be more fit than ancestor, but yeah, we just have to do the experiment at this point.
That's good. It looks like you may have questions in the chat. Do you want to click there?
Oh, okay.
Take one more from the chat if there are.
I think I have to go out of presentation mode for that. If at all. There we go. Oh. Thank you. That's great. Very flattering. Thank you. You can use this if you want.
All right, so we're moving from a very finicky organism to arguably an organism that is exquisitely good for molecular biology and functional studies. And I think Audrey will give you a very good snapshot into how many tools there are in the fruit fly, Drosophila melanogaster, and how she has really developed a whole suite of tools to address some really important questions. And so Audrey's fantastic, but in addition to the awesome science, which I will let her tell you and which should speak for itself, I also want you to appreciate her great artistic abilities. So she she makes beautiful figures. Slowly my talk is now all just her figures. And then I went down to the microscope room and looked, and she had drawn like little cartoons on Post-it notes that are hanging on the wall. She also, I shouldn't blow my wad until she defends, but she also changed our whole lab. Now each room has an animal theme. And that is all thank you to Audrey. So there's many, she's multifaceted and very talented.
Thank you. All right, check. I sound good, audience? All right. Well, thank you, Melissa, for that great introduction. And I just want to say I'm pumped to be in person. It's great to see actual like physical faces and like doing things that we kind of did in the before times. So thanks for coming out. And I want to thank everyone on Zoom who's also tuning in right now as well. All right, so I'm just going to take a step back and take a really paint a really broad picture of the fundamental questions that we're interested in answering in the Harrison lab. And that is, all animals arrive from two specialized differentiated haploid cell types that will eventually merge after fertilization and develop into a full-blown adult. However, before that can actually occur, the embryo has to undergo a process of reprogramming in order to remove all the parental signatures left behind by the haploid parental cell types so that the embryo can reach a state of totipotency. This is particularly critical because then that embryo can form all the cell types required to make an adult multicellular organism. And so we in the Harrison lab are particularly interested in this time in development, mostly for studying the reprogramming factors and how they affect transcription. So we can investigate fundamental mechanisms of gene expression at this time. And so, of course, a barrier to transcription is chromatin itself. All us in this room know that DNA is not just free floating in our nuclei, but instead is wrapped around nucleosomes made up of histone monomers. And so the accessibility of DNA is actually an important factor in determining whether a transcription factor can bind and access its cis-regulatory regions to induce gene expression. However, a factor that can actually overcome this barrier is a specialized set of transcription factors called pioneer factors. And what makes these so unique compared to other transcription factors is their ability to bind and displace nucleosomes to induce local chromatin accessibility at cis-regulatory regions, which can be really important because other non-pioneer factors can then take advantage of this newly induced chromatin accessibility and activate their gene targets. And so a really broad question I have for my thesis is, how is this pioneer-mediated chromatin accessibility maintained throughout early development, especially when we see these rapid reprogramming of transcriptional networks? And so pioneer factors are not the only mechanism of inducing accessibility. I think we all know that chromatin itself can be directly modified. We can either add modifications to the histone tails that induce heterochromatin and are repressive, or euchromatin, which is more accessible and allows the recruitment of transcription factors to those binding sites. And so I'm going to be focusing on specifically histone acetylation, which is broadly a known marker of an active gene. And so the mechanism in which acetylation can actually induce chromatin accessibility or promote it is twofold. And so the protein that actually deposits this mark is known as a histone acetyltransferase, or a HAT. And a HAT can either deposit this mark, in which it creates an imbalances of charges that makes the DNA less tightly wrapped around the nucleosome, so it's a little more free and loose, or it can act as a binding site for nucleosome remodeling complexes to come in, bind to acetylation, and actually eject the full nucleosome from the DNA, thereby making those cis-regulatory regions more accessible to DNA binding factors. And so to turn back to reprogramming, reprogramming actually occurs during a time called the maternal-to-zygotic transition, which is a bit of a mouthful, so I'll be shortening this to MZT. And so this time point is actually characterized right after fertilization, in which the genome is actually completely transcriptionally silent. And so the way the mother actually overcomes this transcriptional silencing is by depositing in her oocyte prior to fertilization maternal mRNAs and proteins that will actually upregulate zygotic transcription. And so as transcription in the zygote is turning up, those maternal factors are slowly degraded over this time point until we reach gastrulation and the MZT is complete and the genome is fully activated. And so in humans, this process of the MZT occurs and zygotic genome activation occurs over about 3 days. But this is a conserved mechanism. And so in fruit flies, it only happens in 3 hours. And so of course, in the Harrison lab, we like a nice speedier way to do our science. And because this is a conserved time, we use the fruit fly to study these essential transcriptional changes during early embryogenesis. And so to really familiarize the audience here with the Drosophila-specific MZT, it actually occurs over 14 really quick nuclear cycles. In fact, these cycles are so quick that it blacks gap phases entirely and instead switches between a phase of replication and mitosis for these first nuclear divisions. And so we use these 14 cycles to sort of tell time through development and at what stage in the MZT we are at through 1 through 14. And to kind of reiterate what I mean by that, transcription starts around nuclear cycle 8 right here at very low levels. And by the last cycle, nuclear cycle 14, it's almost pretty widespread until the MZT is complete right after that. And so a really important player that actually initiates this transcription is a maternally deposited pioneer factor called ZELDA. Now, ZELDA is deposited as an mRNA, it's translated in the new embryo, and then it begins to bind and displace nucleosomes to activate transcription at NC8. Previous work by Melissa during her postdoc in collaboration with Mike Eisen's lab did a bunch of ChIP-seq to look at histone modifications throughout this developmental time period during the MZT. And what they found is that ZELDA-bound regions were actually enriched with histone acetylation, specifically H3K18, H3K27, and H4K8 acetylation at ZELDA-bound sites. And so they wanted to investigate this correlation. And so they removed ZELDA from these embryos by making maternal clones. So these embryos did not have any maternal ZELDA present. And they again chipped for H3K18 acetylation specifically. And basically what they found was that without ZELDA, H3K18 acetylation was robustly decreased in these embryos, creating a potential relationship between ZELDA and this histone acetylation. And so this really laid the groundwork for the questions I have in my thesis, in that is there potentially a coordinated process of genome activation between a pioneer factor like ZELDA and a potential histone acetyltransferase in upregulating the zygotic genome? And so really that's what I'll be tackling here first. And so of course we didn't know what potential hat might be working at this time, but we had some pretty obvious clues going into that. And the first being that the CBP P300 family, which is the vertebrate homologs, name of the homologs, are known to acetylate all 3 residues that are found to be enriched at ZELDA-bound sites. In Drosophila, unlike vertebrates, there's only one homolog, and it's called Nejire, which I'll be shortening to just Nej. Furthermore, this family is so prolifically acetylating active gene areas that chipping for H3K27 acetylation is actually a known way to define active cis-regulatory regions in a whole genome context. And so, of course, I wanted to know, is Nej even present at the time during the MZT? That would be a nice indicator that it could be possibly the hat we're interested in. And so I endogenously tagged Nej using CRISPR-Cas9 system to actually look to see if the protein was present. And so what I hope you can appreciate from these images, we're at the last cycle in the MZT, and you can indeed see that there is GFP Nej expression at this time point, indicating that maybe we should investigate this protein further. And so this, of course, doesn't tell me if it's binding at ZELDA-bound sites. And so to actually answer that question, I performed ChIP-seq to look at both factors in the early embryo at NC14. And so what I found is that both sites, both proteins are bound to thousands of sites at this time point. However, I was more excited to find that when I enriched for shared sites between both factors, that over half of the Nej-bound sites were actually shared with ZELDA. pointing to probably that this is the hat that we are indeed trying to investigate. And so I wanted to know, given that NEJ is a cofactor, whether it requires Zelda to be recruited to the genome during the MCT. And so to answer this, I performed Zelda RNAi in with a maternal driver to maternally knock down Zelda in these embryos. So then again, I chipped at the same time point for NEJ to see if NEJ recruitment was impaired. And so I don't have a whole genome analysis of this quite yet, but I have a representative genome browser track where we can start to see some trends forming, in which indeed we do have some shared sites between Zelda and NEJ. But when we look at the embryos that are actually depleted of Zelda at this time point, you can see a significant loss of NEJ recruitment in these embryos. Furthermore, there are some sites that are specifically unique to NEJ that did not have Zelda in the first place, and you can see that they're still able to be recruited regardless of Zelda being present. So this really told us that Zelda is essential to recruit NEJ to the genome at this time. And so, of course, I want to know how this affects histone acetylation, given we see all this histone acetylation at Zelda-bound sites. So again, I chipped for H3K27 acetylation, both in wild-type and Zelda RNAi embryos. And you can see those results here, where shared sites seem to show, again, a robust loss of H3K27 acetylation, where we've lost NEJ. And we have some, it's a little hard to see here with this projector, but trust me when I say we have some H3K27 acetylation at the NEJ unique sites. So really all of this is to say that this data supports the idea that Zelda is required to recruit NEJ to the genome and to deposit H3K27 acetylation at these shared sites. And so I wanted to share some data that was actually not made by me, but made by my lab mate, Tyler Gibson, where he's actually working in S2 cells, which is an insect-specific cell culture system that does not endogenously express Zelda. And so by transfecting and expressing Zelda in this system, he can fine-tune some of the mechanisms of Zelda-mediated pioneer factor activity. And so what he did here is he expressed Zelda, and then he chipped for Zelda to identify all the sites in the genome that are now bound, Zelda binds in the genome in S2 cells. He then performed a TAC-seq, which is a mechanism in which we can identify accessible chromatin areas of the genome. And he divided his Zelda bound sites into two regions, those where Zelda is bound and they have no change in accessibility. So Zelda is not pioneering these regions, and those in which Zelda binds and actually induces accessibility. So these are pioneered sites. And so he was interested in knowing, how does this actually affect the chromatin state, and what does H3K27 acetylation look like in these different classes of Zelda bound sites? And so when he chipped for H3K27 acetylation, what he found was basically there's a little change in acetylation in the sites in which Zelda does not pioneer. But what's exciting to see is that there is an increase in H3K27 acetylation at sites in which Zelda is actually able to pioneer. So together, these data supports this hypothesis and this model that we have going, that the pioneer factor Zelda and its pioneering activity is required to recruit a histone acetyltransferase like NEJ to the genome to acetylate the surrounding residues. Now, we don't know exactly how this is happening, whether maybe it's a direct interaction between Zelda and NEJ, or maybe it's indirectly where Zelda comes in, induces accessibility, and then a transcription factor can bring in NEJ secondhand. Regardless, we know removing Zelda from the system impedes NEJ recruitment, and we get no histone acetylation. And so really the next big question I have for my thesis that I'm actively working on now is, what is the functionality of NEJ during this time point, and whether its histone acetylation is important for upregulating zygotic transcription or maintaining accessibility through this early period of embryo development. And so to answer these questions, I turn to an optogenetic approach. This is a method that our lab has previously used to study Zelda. And basically we use the Cry2 optogenetic system, where we endogenously tag a protein of interest, in this case it was Zelda, on the N-terminus, which is a blue light sensitive domain. And so the idea is you keep your embryos in the dark when you're not interested in inactivating this protein, And then when you treat them with blue light, this causes a conformational domain change in some capacity that impedes protein function. And so my lab mates, a previous postdoc, Stephen McDaniel, as well as Tyler, validated this system in our lab. And so I just kind of wanted to show you how this works and the power of why this is such a cool system to use to study molecular mechanisms. And so basically, they are able to take proteins that have a nuclear fluorescent label on them and then slap them on the episcope and actually visualize in real time these embryos going through the MZT. So they can precisely stage where they are and then turn the blue light on for those specific nuclear cycles. And so I have a little video here that I'm mostly showing out of vanity because I think it's cool. But basically, you can watch, and I have it annotated down here. This is a wild type embryo that's happily dividing through the MZT. You can see the nuclei all divide synchronously. I have a few plane changes here because it's on an episcope, not a confocal, but you get the idea where this embryo towards the end here is actually able to reach gastrulation and move on and develop like a happy little flywood. And so they, of course, wanted to try this with the Cry2-Zelda system, which again, they treated with blue light from NC10 to NC14. And instead of showing you another video, I just have a still here to remind you this is a wild type embryo at NC14. And when they did this same experiment in Cry2-Zelda, it looked like this, which you can tell is not happy at all. We have nuclei that are super bloated, they're starting to drop out, and the embryo will actually not survive outside past the MZT, and it will not make gastrulation. So this is a really cool way to study the function of this protein because we can do single embryo experiments by performing these on the episcope, taking the embryo, picking it, and then processing it for single embryo sequencing. And so we can look at things like transcription by doing RNA-seq and ATAC-seq looking at accessibility. And that's what they did in this paper. So of course, I thought this was really cool. So I wanted to try it on my protein. So I was able to endogenously tag the N-terminus ofNej using CRISPR-Cas9. And I wanted to perform basically the same experiments. So, but before I did that, I wanted to validate the system. So I performed hatching count, which is basically I treated these embryos with blue light for the first 4 hours of embryo development. And then I just wanted to see, do they survive after that to hatch into larvae 24 hours later? And these are the results here. And you can see a pretty profoundly strong result in which only the Cry2 Nej embryos that are in blue light show 100% lethality, while wild type and those left in the dark are relatively healthy. So not only does this indicate that my system seems to work, but it shows thatNej is essential for early embryo development. So the next big question is, okay, it's essential for embryo development, but is it functionally inactive too at this time? How does it affect histone acetylation? And so our collaborators in the Minervic lab in Sweden actually performed this Western blot where they treated embryos with blue light for the first 2 to 3 hours, and then blotted forNej-specific H3K18 and H3K27 acetylation. And you can see that only again in the blue light treated Cry2 Nej embryos do we see a reduction in these acetylation marks. So it does indeed seem that the system is able to functionally inactivateNej when we treat it with blue light. So I wanted to see if we see a similar dramatic phenotype and lethality like we do in Zelda during the MZT. So I mounted my Cry2 Nej embryos on the episcope and I treated them with blue light again to see if we have this similar dramatic death phenotype. And again, you just watch the hypnotic nuclei divide over and over. And what's interesting compared to Cry2-Zelda, and I make a few plain changes here, is that unlike Cry2-Zelda, these embryos actually are able to make it past the MZT. And so we know in the blue, when I treated them with blue light, they don't survive, but they are able, even though I'm treating them with blue light at this time, are able to survive past the MZT. And so this was really kind of surprising to me because I was really expecting a phenotype more like this. And so I kind of had one of those second guessing moments of just like, okay, well, maybe I'm accidentally inactivating zygoticNej because I had them in blue light too long, or So me and Melissa were talking about it and what we should do. And this undergrad that I had the privilege of working with at the time was like, well, why don't we just do like maternal crosses and see if you get the same phenotype? And I was like, yes, that is brilliant. That is the perfect experiment. I love that. So she set up this experiment here where I have the cross up here just because I know not everyone thinks about maternal contribution all the time like we do in the Harrison lab. But the idea basically is she set up these crosses where you either have mothers that are homozygous for Cry2-Neg or completely wild type. And so the idea is if we treat them with blue light, only those that have a maternal contribution that are Cry2-Neg specific will you see a phenotype. Will you see a phenotype? And so she performed this experiment again. And I was super relieved to see that we, again, have that really robust lethality phenotype only in the maternal Cry2-Neg line, indicating that not only is Neg important for early embryo development, but maternally encoded Neg in the oocyte is essential for development as well. So this really led the path to sort of my future directions, where I now have this validated system where I can precisely inactivate Neg across the MZT at time points of interest. And then I can do single embryo experiments such as RNA-seq to ask whether Neg is essential for activating zygotic transcription, or single embryo attack to see how it might affect the loss of acetylation might affect chromatin accessibility. And so, of course, this is a great plug for my rigor and reproducibility, where because this is single embryo experiments, it's really important to have multiple replicates because we want to make sure, because this is a single embryo, that this is actually, happening in every single embryo that we're assaying. So I have at least 4 replicates for everything that I do. A great scientist always has great controls, and one of our controls is using wild type embryos that are in the blue light, so we can assay for any sort of non-specific expression, such as those may be affecting circadian rhythm, or those may be heat shock because they've been on the episcope for a little too long. So we can cancel those out in our Cry2-Neg specific replicates. And then finally, I think the precisely timed extraction where we can actually look at them at the scope and I can set a timer to know how far I am into a nuclear division so that I can precisely align the time points across all my replicates, that they should all have similar either RNA-seq or attack-seq profiles. And so we've reached the end of the talk. So just to kind of summarize what I've shared with you guys today is that a pioneer factor like Zelda is required to recruit the maternally essential Neg to the genome during the MZT. Now, we don't know whether this is through a direct or an indirect mechanism, but regardless, we hypothesize that the acetylation that is required by Neg might be important for upregulating transcription or possibly maintaining accessibility through early development. And although Neg is conserved, Zelda, however, is not found outside of insects. And so what's interesting about this study is even though Zelda won't be found in other animals, other models such as zebrafish have found that pioneer factors are important for recruiting a histone acetyltransferase to the genome to upregulate zygotic transcription. So even though this is not necessarily, the proteins are not conserved, it's a conserved mechanism that fundamentally highlights that this is an essential process undergoing during this time of development. And so with that, I just would like to give a huge shout out to my lab, of course, for Melissa for all her guidance, Tyler and Katie Holland both contributed data to this project. And of course, I want to thank our collaborators as well as my committee. So if there are any questions, I would be very happy to answer them. Yeah.
Awesome talk. I was wondering with the acetylation marks, do you see that they're cleared post-fertilization or do you see any maternal imprinting?
What do you,
like maternal imprinting, they can continue to keep some of the epigenetics.
Oh, right. I see. The maternal, yeah. So no, we don't think a lot. The chromatin is known to be in sort of what's called a naive state, where upon fertilization, there are not a lot of transcriptional, or there's not a lot of histone modifications. And so the ChIP-seq that Melissa did with the Eisen lab saw that they start to come on right around nuclear cycle. I think it's like 8 to 10-ish. So yeah. Acetylation itself is also not really known to be an epigenetic mark. Francisco?
Pretty close. That's a clever answer.
Like, so are you, so in the, when the paternal line is cry 2?
Yeah, does that matter?
She didn't really see any, it looked wild, it looked essentially normal. It didn't seem to matter that much. Oh, I see what you're saying. Sorry. It's also zygotically lethal. So when I put them up for blue light experiments, I was expecting to see lethality. We just weren't sure if it was maternal or zygotic. So I would imagine they tend to die zygotically with around, I believe, embryo stage 9 to 10, which is a bit later. And so I would imagine if I kept treating them with blue light post MZT, I would see lethality too.
And in your acetylation acid, you have maternal coping
And acetylation?
Yeah.
They treated them with blue light during the MCT, and so my guess is that most of that is maternally derived. But I have an idea that maybe zygoticNej turns on maybe towards the end of the MZT, but I don't know how strongly, and it's kind of preliminary evidence. But I would say most of what we're studying here is maternally induced. Cool. Thanks, guys.